सध्या आवाज
Good morning, dear faculty members, research scholars, and students. Welcome to Faculty Development Program on Emerging Research Trends in Pharmaceutical Sciences on day 3. Before we proceed to the speaker introduction, our principal sir will invite our today's speaker, Dr. Vasantharaju, Professor and Principal, Rani Chennamma College of Pharmacy, Belagavi, Karnataka. We are honored to have Dr. Vasantharaju sir with us as our explores this topic, redefining analytical practices using green chemistry, challenges and opportunities. Sir, Manjappa sir. Dear participant, mute yourself. So I request Mr. Anusha Madam to introduce today's speaker.
Good morning, everyone. It is my great honor to introduce our esteemed speaker for today's session of our national level 1 week e-FD on emerging research trends in pharmaceutical sciences. Dr. S.G. Vasantharaju sir is a highly accomplished academician and researcher in the field of pharmaceutical sciences. Dr. S.G. Vasantharaju sir currently serves as the professor and principal at Rani Chennamma College of Pharmacy, Belagavi. With over 30 years of teaching experience at the undergraduate level and 27 years experience at postgraduate level, he has dedicated his career to excellence in pharmacy education. Sir contribution to research is impressive with 18 years of research experience, guiding over 80 postgraduate students and currently supervising 7 Ph.D. scholars with 1 already awarded. Sir holds an M.Pharm in pharmaceutical chemistry and Ph.D. and has to his credit more than 79 publications in national and international journals with an H index of 10. His academic leadership includes active coordination of key accreditation process such as PCI, AICTE, NBA, and NAC. Dr. S.G. Vasantharaju sir has also been a resource person in over 20 faculty development and skill development programs and has participated in 9 international and 15 international conferences, along with a coordinating 10 conferences and workshops himself. Sir has 5 prestigious awards and has contributed to academic literature with 2 books and book chapters. Today, Dr. S.G. Vasantharaju sir will be delivering a session on highly relevant and timely topic in drug development, redefining analytical practices using green chemistry, challenges and opportunities. We are truly privileged to have such a distinguished personality with us today. Please join me in welcoming Dr. S.G. Vasantharaju sir to deliver session.
Yeah. Good morning, all of you. Thank you, Ms. Anusha, for the nice introduction. Shall I share the PPT?
Sir, please proceed.
Is it visible?
Not yet, sir.
Not yet. Now?
No, sir.
Okay. Now it's visible.
Yes, sir. It is visible now.
Thank you. Thank you. Slide share. All of you can see?
Yes, sir. Now it is okay.
Okay. Good morning, all the participants. We'll start with the today's topic, redefining analytical practices using green chemistry, challenges and opportunities. So myself, Dr. S.G. Vasantharaju, Professor and Principal in the Department of Pharmaceutical Quality Assurance, Rani Chennamma College of Pharmacy, Belagavi. I was professor at the same department from the Manipal College of Pharmaceutical Sciences, Mahe Manipal. So I got the invitation when I was there. So recently, just on 3rd of July, I joined this college as a principal. So these are my contents of the today's talk: introduction, history and background, objectives of green analytical chemistry, core green analytical principles, efficiency and waste reduction, trends in GAC, green solvents in industry, application of GAC, advantages, challenges, changing mindsets, and some of the softwares which are used to show the effectiveness of the greenness of the chemistry, which is to be given to the data to be given to the where you are publishing the journal, to the journal you have to give. So a few things I will discuss there. So anything in between the slide goes off, any questions are there, so you are welcome to disturb in between. So introduction. Now traditional analytical methods optimize speed and cost. That means the thing is that innovation, that is need is innovation means, so when you need, we go for the innovation. The need is the mother of all the innovations. If you need, then only we'll go for this one. The concept of the green analytical chemistry came from the green chemistry, because earlier when the concept of the green chemistry came means, so when 30 years back, when we all done our M.Pharm, the synthesis used to do, what we used to do is some of the synthesis to carry out for several hours of the time, with a lot of energy usage, with lot of water for the, what is called the condenser, you have to put water. So after that, the concept of the green chemistry came into picture, like use of the microwave ovens. So domestic microwave ovens they were using, so that 30-40 hours of the refluxation came down to the 4 to 5 hours or even less than this one. After that, several developments came. Nowadays, what is called conventional, these instruments are available to the laboratory synthesis. So this is how the concept of the green chemistry came into picture. Nowadays, nobody will go for the what is called the conventional methods like reflecting for several hours and all those things. It's a few hours job with a good yield, and you can lot of things you can contribute for any environment in terms of energy, in terms of water, in terms of time, all those things. So this, on the basis of this one, the analytical chemistry, what is that earlier we used to do the simple colorimetric methods. After that, spectrophotometric methods came. After that, several other methods came. But when the what is called more and more advances came, so HPLC become part of the analytical system. There, we were using almost what is called acetonitrile, methanol, all those things. These are the common solvents which are commonly used in the HPLC, because most of the drugs what you are using are the either weakly acidic or weakly basic drug. That's why we have to use, compelled to use those agents. The what is called as a mobile phase. So then after that, our green chemistry, which is contributing for this one, why can't we go for in the green chemistry? I forgot to tell about that. The not only microwave, several other things are there. Some of the oils, vegetable oils, they tried for the synthesis. Some of the sonochemistry they tried for the synthesis. Like that, several things are there. But that is not the part of our the discussion here. It is the only green. So this become the green analytical chemistry. The thing is that why can't we go for the green things so that we can reduce all these harmful chemicals, so which are going into the environment. Time can be reduced, and whatever is there, if we use the ethanol, if you use the other solvents, so our environment can be saved from the what is called the chemicals, as well as sometimes some cases it is a reduction in the what is called the runtime, and all those things are there. So then environmental and health impact often are ignored in the traditional analytical chemistry, because most of them are volatile in nature. Most of them, whatever the chemicals, whatever using are, they are causing the environmental effects. Because if you go to the industry, they all know, no question of nowadays availability of the spectrophotometer. Everybody, whether it's analytical method, bioanalytical method, BA/BE studies, whatever it is, they use the higher instruments like UPLC, LCMS, HPLC, and all those things. For that, we need a lot of solvents. You can say it's a that is like they are using like a water, acetonitrile, methanol, everything they use like water, because they inevitably we have to use it. So, but this traditional the analytical chemistry, so divide of this, they ignored environmental impacts, health impacts, the health impact of the what is called the method developer, and the persons working in the laboratory, that is ignored in the traditional chemistry. Then green analytical chemistry aims to reduce this hazardous waste. So whatever these solvents what you are using, so in the academics, fine it is. So something whatever is there, our students say that we use and put it in the sink. But industry, they have to give for the account for this one. What is effluent coming out of this one? The regulatory agencies will ask that. What you will do with the effluents? How do you process it? All those things they will ask. People has to keep the account for this one. It's a several liters of the what is called the acetonitrile and other things are using in these what is called the conventional analytical methods, especially by the HPLC. Now, this green analytical chemistry aims to reduce this hazardous waste, aims for the effect on the health impact of the whatever the user, health effect of the persons working in the laboratory. Then if you go for the history, it's not a just immediately after the whatever the green chemistry came into picture, immediately they did not know why can't we apply. All those things are there. There's a several history. There's a history is there. I'll go in the key milestones of this history. 1968 Biosphere Conference. So they thought of that. Why can't we go for this reduction of these hazardous solvents? Why can't you go for, you can reduce the environmental pollution by using the greener solvents. You can reduce the health impact of these solvents on the user. So that all discussed. Then again, 87, the Brutherland report gave the same thing. So then the 1990 green chemistry movement initiated. So this initiated and came into picture. This detail I will not go into. This is history. So just if you go for this Biosphere Conference, this Brutherland report, all those things, if you give you will get the history, you can read it. So then environmental awareness fuels demands for the sustainable practice. So now that time the journal asking what is the effect of your means how you are going to save the environment. So these are all the hazardous solvents. So what way you are going to contribute for the environmental. So ISO 14000 also asking what is the solvent used when you are implementing. So how do you do this? They'll ask what you will do with the solvents. So what they will do is they will collect the solvent, reprocess it, some other purpose they are using. That's what the environmental awareness fuels the demand for the sustainable practice. Then next, objectives of the green analytical chemistry, evaluate the current practices. So to reduce the hazardous chemicals, what we are doing and to evaluate that current practices, they identify adaptation barriers. So don't think that the method development using the greener solvent, it's not so easy. It's not so easy because most of the drugs, whatever available, you can say 90% are either partly ionized, either too weakly basic, too acidic solvent. In that case, so what is called the development selection of the greener solvents is a very, very big task. That is the adaptation barriers. It's not so difficult if you go for the conventional methods. Just change the mobile phase, composition you change, buffer capacity you change. So buffer you change, then method will be standardized. But it is not so easy in the develop a method using the green analytical chemistry with the greener solvent. That's why one has to identify the adoption barrier in the green analytical chemistry. Then showcase the innovation. What innovation is done? So innovation is done in terms of the green chemistry. So that is, if you use whatever the naturally available the solvents are there, it won't affect the nature. It won't affect the human being which are using these chemicals. All those things will not be there. So that you have to show that the green analytical chemistry showcase these innovations. Then promote best sustainable practices. Good laboratory practices is the old concept. GLP is the old concept. Now is the best sustainable lab practices. How much of the solvents, so you are using, what chemicals you are using, what it contributes for the nature, how it harmless to the nature, how it harmless to the person. So one has to, in addition to the common practice, the good laboratory practice, one should have the best sustainable lab practices. These are the objectives. So then core green chemistry principle is that if the prevention to the what is called what aim the green chemistry principle came into picture, green analytical chemistry came into picture. So the prevention is nothing but prevention to the atmosphere, prevention to the nature, prevention to the ourselves. That means the persons working in the laboratory. That is how it is preventive. Second one, sorry, something, the atom economy. Can you hear me?
Yes, sir, it is audible.
Yes, okay. Atom economy. So what level chemicals you are using, to that one, what economy it is. So because you have to, when you go for the analytical chemistry means, so you cannot use simple laboratory chemicals. You cannot use simple, what is called the AR grade chemicals. We have to use the HPLC grade as well as LCMS grade chemicals. So in such a case, what is the atom economy you are going to use? Less hazardous synthesis. So it is the principle of the chemistry is nothing but same thing. So synthesis here also, it is nothing but the chemistry part only, how hazardous it is. Energy efficiency, microwave has assisted. Some of the reactions are using microwave in the synthesis use. But here also same principles can be used. Safer solvent and auxiliaries, replacing toxic organic solvents is the safer solvent and auxiliaries. It's total in general green chemistry. Few of them is adapted into the green analytical chemistry. So then renewable resources. So ethanol if you use, water if you use, it is all renewable resources. It is renewable. Lot of resources are there for us, ethanol. So you can get it's naturally available. So it is compatible with all the natural creatures. So then lot of resources are available, but other chemicals, like you have to go for the chemical synthesis, to synthesis, lot of processes is to be used, lot of steps to be used, a lot of purification steps to be used. So that's all that is renewable sources if we use, so that problems will not be there. Then designing safer chemicals. So not necessarily water as well as whatever the ethanol and all. Several other things like some of the oils can also be used in future. So far, no things are not available. Only whatever the methods are there which are available based on the ethyl alcohol, some modifications in the buffer. Only that much is so far literature reviews are available. In future, so if the research continues, so that leads to say maybe availability of the more of the safer chemicals, more of the what is called the less hazardous solvents, more of the safer solvents, more of the they may get from the even renewable sources. They will be compatible with this one. That is the core of the green analytical green chemistry principles. Then just continued reduced derivatization. So in this case, this is green analytical principle. So several steps can be reduced if you go for this green chemistry. Catalyst with use of the more and more catalyst, so you can reduce the time. So design for the degradation, biodegradable compounds, safe disposal. If you use the ethanol and all, if you synthesis, okay, several chemicals are there. So if it is a biodegradable, it's easy to dispose of. Here in the analytical chemistry, so if you use the biodegradable chemicals, so what is that? Their disposal will not be a problem. Again, if you use the conventional solvents, the disposal is another problem. So one has to spend a lot of what is called the money to dispose of such chemicals. But if it is a biodegradable chemicals are there, biodegradable solvents are there, their safe disposal, you can what is called, you can surely say that. So regulatory agencies will not ask that. So what is this one? If it is a safer one, if it is a biodegradable compounds, then real-time analysis, process analytical chemistry is possible with this. It is also possible with the real. It is also possible with the conventional solvents, but real-time analysis, that is a Ph.D, Ph.C, that is process analytical chemistry, is also possible. Safer chemicals for the accident prevention. That means you are working in the working place, if you are working in an industry, if you are working anywhere, one has to pass the OSHA guidelines. So OSHA toolkits. So that this OSHA toolkits or OSHA guidelines will tell about the workplace safety. It did not say about what solvent use, all those things. How safer your workplace, how safer your work persons. That is, if you use the safer chemicals, so accident prevention will come to, so what is called, to lesser extent. If other highly inflammable chemicals or more volatile solvents if you use, so that may lead to a lot of accidents. The same, so if it comes under the OSHA toolkit or the workplace safety, whatever our real this one biodegradable chemicals are safer enough. So then reduce the derivation saves a lot of material. This what is core principle will go for the analytical the synthesis chemistry. Then catalyst, less waste is there. Reusable catalyst can be used in case of the synthesis. So real-time analysis improves the process and safety. So then, these are the, in general, the green chemistry principles. Out of this one, few can be adapted to our analysis, reduced time. So then real-time analysis, safer chemicals for the accident prevention using the OSHA, the toolkit, workplace safety, biodegradable chemicals are there. Few of them is taken as the green analytical chemistry principles also. So what about the green solvents in industry? So these green solvents can be used for cleaning, extraction, as well as synthesis. So it's not necessarily only for the synthesis. It is not necessarily only for the analytical methods. These can be used for the cleaning purpose also. Extraction of various constituent from your formulation, from the natural source, or from the biological matrix. It can be green solvents can also be used for the synthesis. These are all used benefits, lower toxicity as well as biodegradability is there, techniques, that is, these are all the ATP, MEA, UAE, these are the different techniques in synthesis where analytical techniques can be used. So application in HPLC, these greenhouse solvents. So that is, there is a one table is there in the next slides. Few of them I will explain here. Zonisamide, ethanol and water is the solvent for this one. Successfully, they are developed and validated using ethanol water. This using this the what is called the for this drug, as well as published. Atorvastatin, those statins are not now commonly using. Then acetic acid and ethanol is one of the solvent. So then the acetylopram, acetylolom, ethanol plus buffers of suitable pH can also be used. Some more is there in the next slides. When you go, we'll see there. Advantages: reduce hazardous wastes, enhances safety and efficacy, support sustainable chemical manufacturing. You can reduce the several hazardous waste. You can go for the safety and efficiency because they are not harmful to the chemical. Continued inhalation, continued work with those the hazardous chemicals will lead to several the health principles by using these solvents. But when you use the biodegradable solvents, the obtained from the nature, so that will safety as well as efficiency is there. They support sustainable chemical manufacturing. Their manufacturing will have a few steps, either simple extraction and simple purification, as well as disposable everything is there. But I told in the previous also, it's not so easy that so you cannot simply with whatever the available, you take ethanol, you take water, you take acetic acid, you take different buffer solutions, you can develop. It's not so easy. There are several challenges are there. What are those challenges? High initial cost. Okay. So several trials have to be used. The ethanol, whatever is there. So all of you know that the ethanol purity is also very important. Whatever conventionally available, the ethanol cannot be used. What is available, the industrial things, it cannot be used directly. It is to be what is called free from water. That is super dry. When you use the super dry, you have to be very careful. So suitable environment is to be prepared, because as soon as you open this bottle, this ethanol will immediately absorb the moisture and water content will be increases. So that is another thing is that so you should make some arrangements to use these chemicals at a suitable environment. Limited green substitutes. Nowadays very green limited green substitutes. Some of the buffers are there. Some of ethanol is there. So water is there. Limited. Maybe this challenge can be overcome. So with the invention of more and more of the what is called the greenhouse solvents, which is obtained from either vegetable source or animal source, may be a challenge. It is again a challenge. So then gaps in the regulations and training. One should have the proper knowledge. One has to convince that how this method is better to the, and lot of training is to be given. So simple, those who have worked so 2 months, 3 months with the HPLC and with the conventional methods, if the new drug if they give based on the pKa and the properties, all those things, immediately within 1 or 2 days he or she can develop the method. But using the greener solvents with the these solvents, it's very difficult. A lot of training is required. So we should have a regulatory things for also. So regulatory persons, you have to explain how better this one, how all those things to be explained for them. Then lack of standardization, maybe based on again the availability of this natural resources solvents. So it requires a standardized source, as well as the standardized purified method should be there. That's why the first point, it suits that high initial cost. So, but in long run, with respect to the environment, already our environmental is polluted lot. Groundwater, lot of chemicals we are encountering. So then other things are there. So because of the lack of standardization, So maybe after the few research, so one can what is called in future. So this standardization will be what is called optimized, and it reduces the initial cost, and maybe availability of the more and more greener substitutes may be available. And after usage of these solvents, so it requires some, you may get more and more trained person to train the newcomers. Then challenging mindset. Public and scientific community support the greener methods. So that means easily available. All chemicals or conventional methods are easily available. So the what is called the scientific community should accept these greener solvents. These greener methods should accept. They should understand. So what is the potential benefits of these greener methods? How far in long run, what is the use of this one? So some industry, if they say no, it's a initial cost, procurement of the chemicals, all those things, very few suppliers are there. So they not meet the demands of the supply. So in such a cases, what happens? May not support for these greener solvents, greener methods, because nowadays many are there for the supply HPLC grade and the other grades of the acetonitrile and all those things. But so more and more things are available. So public should understand, scientific community should support the greener methods. That's why nowadays the scientific community has come to the, so what you call such a way that if the method is your greener method, if you apply this greener method to the new what is called the drug development and validation, so they accept faster than your conventional methods. That is what the scientific community so gives the assurance now. The public methods, if your title says that is a greener method using green analytical chemistry approach, so then you just develop with that greener solvents. It may take time. It may take lot of time and other things. And so lot of tools are available. I'll be discussing those tools in future slides. With those things, so if you submit that paper, so definitely acceptance rate will be better and more so than your conventional method. And so that is what the scientific community supports for the greener methods. Then researchers are seeking eco-friendly reagents. Nowadays, because of the environmental pollutions, each and everything they are asking, so what is your environmental contribution? If more of the solvents means how to dispose of the solvent. What is the effect on the environment? What is the effect to the earth? So what is the effect on the person working? So that's why researchers are seeking eco-friendly reagents. More and more they are asking, okay, it is safer for us. So it saves a lot of energy. It saves a lot of chemicals. So it is eco-friendly. So disposal of will not be a problem. So that's why researchers now going to what is called the seeking the eco-friendly reagent. It is a positive sign for the green analytical chemistry. Green analytical chemistry is reshaping research and industrial practices. So maybe immediately after this green chemistry, so they were all doing in the synthesis research level. They were doing the even conventional microwavers, domestic microwave ovens, and suitable what is called the designs are developed like to do the laboratory scale things on. Then immediately after that research, they came into laboratory. Now some of the undergraduate experiments are doing with these microwave things. Each and every small college is acquiring this one. In the same way, the GAC should reshape the research and industrial practices. That is all possible if and more the researchers seeking for the eco-friendly reagents, they work on the eco-friendly reagents. This is what the challenging mindset. These are the challenging mindset for the green analytical methods. So the contest is that, so the nowadays more and more green analytical the practices are publishing more and more green analytical techniques using the HPLC and other, so they are publishing in this one. So maybe like the green chemistry. So nowadays all even the undergraduate level everything they are doing experiments using this one because of it saves time, it saves a lot of chemicals, environmental freely, the hazardous effects are not there like that. So maybe another 5-10 years. So all your analytical methods may be using the green analytical reagents. This table shows that the what is called the several green solvents used in HPLC and other analytical agents, that is zonisamide, ethanol water is application in HPLC, atorvastatin calcium, 5%, 0.5% aqueous acetic acid ethanol for HPLC, etazolamide and acetyloprom, ethanol and phosphate buffer in the HPLC, lavimavudine or zidovudine or neodine, all these things, the viodines using ethanol, it reduces the column diameter. So that is how this is also one of the, not only using the solvent, it is also using the, it changes the column. That is also work is going on that one. So then another thing is that benidipine hydrochloride and chlorthalidone, the combination, ethanol and potassium hydrogen orthophosphate, HPLC, atorvastatin and another one, ezetimibe and fenofibrate, ethanol and 0.5% triethanolamine in water, HPLC method. So molnupiravir, ethanol and phosphate buffer, it can be used for UV as well as HPLC. Betamethasone and calciprotein, ethanol and phosphate buffer, RPHPLC. Like that, you can find a lot of articles. So if you go for the Scopus, if you go for the Web of Sciences, if you give the green analytical methods, so keywords, you will get a lot of things in the net. Softwares and tools used to assess the greenness. Nowadays, the thing is that I have used the ethanol. So my ethanol and water is used. And so the thing is that it reduced the runtime. So it is a eco-friendly solvents. All those things. So how you can approach the scientific community? How you can approach? Simply ethanol water is developed and method is developed and validated means, see, your publications will not be accepted. Your publication will not be accepted. It is accepted if you suitably represent the what is called the eco-friendly nature of your method. Eco-friendly nature of your method. For that, several tools are used, several the softwares are used to assess the greenness of your method with respect to the time, with respect to the environmental effect, with respect to the greenness of the solvent, all those things. So you have to feed your data into the software and tools used, and you generate some of the so graphs or anything. So that data, it is to be a supportive data for your already method developed and validated. If you submit into the, so what is called the journal. So based on that one, so they will accept your paper. We'll see around 3 are there. Many are there, but these 3 I am highlighting. There is one called AGREE, that is Analytical Greenness Matrix. One of this is the AGREE. So this AGREE is a comprehensive tool developed to evaluate the greenness of analytical methods based on the 12 principles of the green analytical chemistry. Numbers is there, that is safe, then environmental friendly. So then safer to the environment, like the 12 principles are there. Based on the 12 principles, this AGREE the matrix is developed. So it will evaluate your greenness of the analytical method based on the 12 principles. These 12 principles, they fed into the software. So you feed one by one ethanol. What is the conversion used? What is your runtime? So what is your RT? Based on that one, it will give the score. What are the key features of that one? There is a visual output. A circular pictogram with 12 segments, each representing the GAC principle. It looks like the pie diagram, a circular pictogram things. It has a 12 segments, each representing one green principle of the GAC. Then each segment is color coded. Green is more compliant, red is non-compliant. One by one data you feed, each of the segment will acquire a color. If out of these 12, how many based on your data, how many are green, how many are in between color, how many are red in color. Based on that one, you can assess your greenness based on this one. The pictograms of the 12 segments of the GAC principle. So they say that based on how many, these many greens are there, this many intermediate colors are there, this is red color is there. If more and more red color there, your method is not green enough. It is not, I told no, simply if you use the ethanol and water, you cannot say green. There are other things are there. So that things, so you have to feed into this one. So then the agree software, and it will give the color coded pictogram based on the different colors on this one. So it will give the how greener method yours. Then final score is ranging from 0 to 1, with 1 being the greenest and the red is the 12th one. 0 to, so what is called again point is there, in between point is there, 0.1, 0.2, point up to 1 is there. So greener one you are getting the highest score of 1 means it is the greenest. How it looks like this. I think if you give this agree software, your Google wins, several pictures will come in that one. So that several pictures, each and every one, so using the software you can develop. So a few of them I have taken here. So in this one, so I think it's very difficult because small one. The first one is that green analytical chemistry matrix for the evaluating the green of the analytical procedure. See all the 12 principles it is given. And here is the score is given from 0 to 0.0 to 1. See that your total score of this one, the B1 says that your total score of the method is 0.37. See here that one, the 6th parameter is green in color. So then 2, 4, 5, 7, 8 and 12 are yellow in color. 1 and 11 are orange in color. But 3 parameters are there, 9, 10 and 3 are red in color. So based on that one again, your evaluator, what is called when you send it for the what is called the publication. So based on this one, what is the score of this one? So they fix the score based on this one. Suppose if it is your score is 0.5 and above, so they consider as okay, greenness of the method is there. There is a good green analytical technique based on this one. So this is how you can take the pictogram by feeding into this one. So other things one, it's very what is called the around. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 are there. So different parameters, different pictograms is there. So it is very difficult to go individually. So because when you feed some data into the agree software, so then you can enlarge and see. These are all the what is called which is taken from the Google with the agree software, agree things. So second one is B, I explained it. You see here it is very difficult to explain. So you can go and you can do it. Another one is that, so one more method is that, so again, one of the score is 0.57. So here is that around 5 parameters are green in color and 2 parameters are red in color. So 3 parameters are orange in color, 2 parameters are, your score will be increasing when green and green is more green principles are there. So this you have to data feed into the agree software. So then you take the pictograms, put it in your research article, and you can communicate them based on the journal principles. They will take, say, whether you can take your method or not. Again, this is one more with the 0.72. So these are all the what is called the pictograms given from the various publications. It is better 0.72. It is like that, say 2, 4, and again it is green, light green, dark green, everything is there. So that is 2, 4, 6, 9, 10, 11 are little dark green, whereas 5 and 8 are light green in color. 7 is the orange, little orange in color, yellowish orange. 1 is yellow in color. Only again red also there is 3 is dark red as well as the 12th is a light red. So based on that one, whatever the data you are generated, you fed into your software. Based on that one, it acquires automatically color, pictogram acquires, and center it gives the score. If the score goes more and more towards the 1, So that means that so your method is more greener. So that means, see, now you I think you got the idea that so what way all we have to helpful, what all the things you have to do, runtime. So then the solvents used, all those things. So you have to optimize. When only then only. So once you develop that one, fit into this one, check the score. Again, you change that one. So fit into this one. Here, when your data going near to one, so then so that is better acceptance rate. Advantages, easy to interpret, simple it is. So they are given score 12 GSE principles. Out of that two principles, so just software is there. What is the first parameter is asking? So what is the solvent used? All those things. Feed one by one. Automatically, when feeding all the data, you will get the score from 0 to 1. So based on that one, so it is the interpretation will be easy. Then you will come to know whether you are getting 0.34, 0.5, 0.8, 0.9, like that. It's easy to interpret. It covers all the aspects of the 12 principles of the GAC. Useful for comparing multiple methods. Okay, so your method. So I have tried all these methods. So this trial gives this much of the, so what is called the pictogram with the score of this one. And earlier method also, whatever the method is there, this is not green principle. So 0.1 is the score, GSC score is there. My method is 0.6 green score. That is more is better. Even within the greener methods, greener analytical methods, you can score, you can give your projection to your method. It is useful for containing the multiple methods. Examples. Etizolam and acetylopram method was scored 0.78. These are the reported methods which was used score give a 0.8. Beta-methasone and calciprotein method gives a score of 0.89. These are the some of this one. You will get a lot of examples from the research articles. Then ideal for researchers and analysts who want a quick visual and quantitative assessment of their methods sustainability. It is a sustainability. How sustainable method is there? Environmentally sustainable and the eco-friendly things are there. Then it is simple one. Just feed the data, take the score. So then you can say that. So in earlier, what you used to do is, so the method runtime is 20 minutes. I have reduced the method to 10 minutes. The RT is 5 minutes, whereas the old RT is 7 minutes. This is how earlier we used to compare. Same way with the change, with lot of the permutation and combination, more and more of the green solvent you incorporate into the mobile phase and other parameters. So more and more of the greenness, the score you are going to get. So that's why it is ideal for the researcher. The software, the Agri software is ideal. Second one, analytical echo scale. This echo scale is a semi-quantitative tool that assigns penalty points based on the environmental impact of each component in analytical methods. Based on the, it is somewhat different. So that is how greener methods, how greener principles you are used in our analytical methods. On the basis of that one, Agri software was giving the score. But here what is that? It assigns the penalty points based on the environmental impact. So penalty point means so more and more of the what is called the pH you are used. What is this effect on the environment? Based on the more and more of the what is called the salts you are used. So that is how it is affecting the environment. Based on that one, it will give the penalty points. In other way, they are assessing the eco-friendliness of your analytical method. Scoring, it is starts at 100 points. Penalty points are subtracted for it. That means first you score with the 100 based on what chemicals you use, what combination you are used. So how many salts are you are used in preparing the buffer? So then how many what is its combination? What is its pH? Based on that one penalty point. So first thing is 10 penalty points. 90 is from the score. Your score will reduce. It is use of hazardous solvents. If you use hazardous solvents in the method, so that gives some penalty for some reason. So all the cases, all the way you cannot use the only eco-friendly solvent. Some of the solvents you are using for some other purpose, you are using hazardous solvents. If you use the hazardous solvents, if you feed for that one, it reduces your point. High energy consumption. Again, it's not necessarily your greenness of the method, not necessarily based on only eco-friendly solvents. So what is your instrument used? How much runtime it is? How much energy it consumes? All those things will be fed. That comes to the scoring. Waste generation. How much of the waste is generated in this? That means your runtime is more. So automatically more and more of the waste is generated. So then your runtime is less. Your runtime earlier, some method, greenness of the method, the GAC method was developed for a runtime of 20 minutes. So then you reduce the RT of some 12 minutes, 15 minutes. So you have adopted some of the green analytical technique principles to reduce the runtime to 10 minutes, and your RT will be below. So what is called the 10. So then your waste generation will be less. What you will do with the, so it's you can say that we are using the green analytical, but it is not mean that we can ethanol you are getting as a eco-friendly solvent, you can use it. But the thing is that their extraction, their resource, their purification, their storage, everything it takes into consideration. So that means anyhow it is eco-friendly. We procure from this one, we use it, and if you discard it from the nature, it is not harmful to the nature. But what is the cost of the chemical? So what is the purification? If more and more solvent you are used, so its cost increases, your method cost increases, waste generation increases. On the basis of that one, it reduces the, so what is called the score. Toxic reagents, any toxic reagents you are used in the preparation of this, in the development of the method of this. So what toxic solvents you are used? So you have to feed the data, because in the method development you have to give the method. These are the chemicals are used. So this is the things are used. So then, so the publisher or the scrutinizer or the reviewer will check. Simply you cannot say that I have used this, my score is not like that, but he will check whether you are really, his greener method is there. So based on that one, so the penalty points are subtracted by this method using analytical eco scale, like use of hazardous solvents, high energy consumption, waste is generated, what toxic they were from the 100, the scores will come down. Then how to interpret is that more than 75% your score you are getting. When you, as soon as you enter, it is your software is showing the 100 points. Some data you enter, 5 points will be reduced. Second, you add another chemical. So this is the runtime it is used. Another 5 points will be reduced based on that. But after all these things, score reduction, you will get one score. Then how to interpret this one? If your score is more than 75, it is considered as an excellent green method. If it is in between 50 to 75, it is an acceptable green analytical method. It is an acceptable green. One can accept. But again, here one. So you are all going for the publication in the quarter journals, Q1 journals, Q2 journals, Q3 journals, Q4 journals, high impact journal, medium impact, low impact, all those things you are going, publications you are going. Based on that one, again, so your method is excellent, your score is more than 75, 85 is there. There is a chance of accepting your, what is called the publication in the high quarter journal, high impact journal. So then your score is between below 75, but below 50. So they may say that, no, we cannot accept it because so your green score is less. Your green score is less. That is 52. If you use this software, if you are getting 50 to 75, it is an acceptable score. It's less than 50 you are getting, it's a poorer, greener solvent. It's a poor greener method. Greenness is less, very less in this one. No public, no what is called the good journals will accept your if the score is less than 50. Again, between 50 to 75, more than 75, it is near to 90. It's considered as an excellent green method. So without any difficulty, they will accept it. This is how you can do the interpretation using this. Advantages, simple and transparent. Nothing. It's just feed the data. What chemical you are used, what is the quantity used, what is the runtime, so what is its RT. So all these data you just feed it. So you will get transparent data. Nothing like that because those anyhow you method you are using. Methanol is used. This much pH is used. Everybody will come to know this pH is used. Means what chemicals you are used. You cannot hide those data. Can hide those data. So that's why just feed the data, you will get the transparent this one. Then the encourage method optimization. Again, in this one, if it is 50 to 75 you are getting, you want to publish in a good impact factor journal, good quarter journal. In such a case, you can still go for the changes in the method. Once it is changed, you can go for the method optimization. Examples. Zonisamide method scored 91. Atorvastatin calcium method scored 90. These are the already the published methods. So just from the literature I have taken and I have put the score. Then benidipine hydrochloride and chlorthalidone method scored 93. So I can say these are the excellent methods. Excellent methods. Best method for developers who want to quantify and improve the environmental profile of their procedures. Just feed all the things. Once method is developed, you are getting good RT, good runtime within less than this one. So then you feed the data, check the score. If he is getting near to your excellent, what you can do is, so you can still refine your method. So check the pH, all those things, several permutation combination you can make, still you can make better. So you can make your environmental profile of your procedure. That's called the environmental profile of your procedure more and more good. So this is how the what is called the greenness. Actually, this in between that score is there. So we cannot see the something. The first one is some 0.61 from that data it is taken, and after that in between that, so they have detected all those things. This in between that score is there. So if you Google it that one, so you will get the what the type of the what is called the interpretation things you are getting, you will come to know. That's what penalty reagents. Dichloromethane if you use four penalty points, 4 will use. Methanol if you use the 6 points will be. Chlorofluoro then 6 will be this. The instrument energy used, your waste reduction is optimal, your occupational hazard is 0, energy used somewhat, you will all these things if you optimize, so you will get 0 score. But these are the harmful chemicals somehow, maybe for solubilization purpose, other purposes you are using. See, most of the drugs are soluble in methanol in such a cases. So this method is 1 mg per ml is prepared in methanol. Then you have to feed this one, your penalty score will come. So this is how the penalty points are calculated using the software. Another one is the TOPSIS software, that is technique for order of preference by similarity to ideal solutions. That is a extended of the TOPSIS. Is a multi-criteria decision-making tool used to rank solvents or methods based on their closeness to their ideal green solutions. See what solvent, it will give the rank solvents, or methods also it will give rank, which is closer to the ideal green solutions. It will give the rank based on that one. How it works? Each solvent is evaluated against the multiple criteria like toxicity, biodegradability, cost, etc. What solvent you are used? So don't think that the other solvents, each and every, whether it is available from the natural source, whether it is applied from the synthetic sources, it has its toxicity. It has its toxicity and a biodiversity. And what is its biodegradability? How much time it takes to biodegrade in the soil? What is the cost of this one? The cost includes the extraction procedure, purification procedure, everything. Other parameters also there. So this is this property that is against the multiple criteria. Based on that one, it gives the green score. Then the method calculates the distance from the ideal and negative ideal solutions. Whether the ideal solutions, that same ideal solutions like ideal solutions and real solutions. So it will give the how they are distance from the ideal as well as negative ideal solutions. That also it will give the score. Solvents are ranked based on their relative closeness to the ideal. If they are more and more to the ideal solutions, ideal and real solutions, you must have studied in the physical pharmacy, ideal solutions, real solutions. So they are in physical pharmacy, you studied. But the solvents are ranked based on how much they are related to the closeness to the ideal nature, they score better. Based on this one, they against the multiple criteria like toxicity, their biodegradation, their cost, some other parameters they will be collected to check the ideal to the green solution. Then the distance from how it is, distance from this one, very near to the ideal, very near to the non-ideal, the negative ideal solutions. Then those solvents will what is called will come to the near to the green solutions, which are closeness to the ideal. Advantages. Yes. Advantages of this method is handles complex decision making. Several parameters are there. This parameter will taking to come to the conclusion that how greener is your method. So it is a complex decision making will be taken by this one. Can integrate qualitative and quantitative data, both qualitative data as well as quantitative data. Also, you can integrate using this one. Useful for comparing multiple solvent system. This is made in such a way that you can compare the multiple solvent system using this software. Examples. Used to rank dispersive extraction solvents for mepistone in water samples. So this extraction of this from the various in the water samples, how it is extracted, what solvent it is used, based on that one. So this method can be applied. Ideal for researchers and regulatory bodies needing a systemic comparison of solvent alternatives. Based on that one, near to the idealness, near to the toxicity, near to the greenness of the solvents. Based on that one, researchers and regulatory bodies, so they can compare the various solvent alternatives, what better you can use, what source you can use. So that all can be done. The conclusion of the talk is that GAC provides an essential path towards the sustainability. If more and more greener solvents are there, so our environmental will be protected. Okay. Now environmental agencies are becoming strict day by day, So what you will do, your effluent. So how do you what is called the effluent? So industries has to now give more and more. They are spending a lot of time for the effluent treatment procedures. It includes the effluent from the production. It includes the effluent from the analytical, what is called the departments. So they spend a lot of time and they put more and more strict on the visions. But so the days are not far away. The environmental agencies will come to every college, will come to every research institute. They can ask you the suitable data for their disposal. This because day by day the environmental the what is called agent is becoming strict and strict. Don't think that so anything you can do it. But if one can provide, one can use this green analytical principles, chemistry principles. So it is a essential path towards the sustainability. So we also safe, environment also safe. So that is the conclusion. The barrier remains, but the innovation continues. Getting the ideal greener solvent is a big task, a big task. Now what is available? Ethanol. So other things that, so in future I told you we may go to any of the solvent which is obtained from the nature. So that what is called barrier is there. So only one solvent is there. How can be? It is impossible. All those things you cannot sit only on the one solvent. Innovation to continue with the green analytical solvents. So you may get better solvent. That is necessity is the mother of all the innovations. Education, regulations, collaborations are key to adaptation. Should have more and more as to educate the environmental effects of the various solvents. It is to be, now it is, of course, it is teaching at the undergraduate level and it's even the other levels it is that. So the students, when they come with such students, so they will think of, okay, it's harmful to our nature. I have to develop something. Then regulators will be putting pressure on that one. Okay, you cannot use this method. We will not approve your analytical method if you use the conventional solvents because it is more toxic for the environment. It is more toxic for the your what is called that is more dangerous to this one. So then, if more and more collaborations, if you do, so one can develop the better green analytical methods. This is the future of the analytical chemistry. I can say my conclusion, the future, what is the future of the analytical chemistry is the green analytical chemistry. So that is, there is a barrier of course, but if they continue innovation, so definitely that will, where this innovation should start at the education level, in the school level, in the college level, if it is taught, so then only so you can have, so what is called the it will come to the student mind that so it is things that I have to do something for the environment. So then only then your regulatory things will be maybe stricter and stricter. So then there is no what is called there is no alternative. One has to go for GSE only. So this is all possible collaboration with the good research institutes and all those things. That is the conclusion of my talk. Acknowledgements. I am thankful to the management, Vasantidevi Patil Institute of Pharmacy, Kodoli, for giving me the opportunity to share my thoughts with the e-FDP participants. So then I also thankful to my management, Rani Chennamma College of Pharmacy, Belagavi, to give facilities to conduct this one, means to have internet facilities and the suitable amenities. I thank to Dr. Shyam for the excellent coordination. He only contacted me. He only given all these things. So he was in continuous touch with me to share all these things. So then I acknowledge my PG student. She was at Manipal College of Pharmaceutical Sciences. So she collected many data along with my data, and she gave together we actually we collected all the data, nearly around 15 articles, 50 articles, 50 articles. So out of that one, this is the extract of this. The presentation is extract of this 50 articles. So my student Kajal helper, I thank to my student Kajal. Then I really the gratitude towards my respected audience for their patient listening, and thanks for all. References, around 50 references are covered. This is the extract, the talk is extract of this one. Around 21 slides is the summary of that 50, or nearly 50 references. Out of that one, around 10 I have put here. So these are 5 in this slide. So remaining 5 in this slide. Then Thank you very much for one and all. If you have any queries, at that time, if I have the answer, I will interact with you. Or else if you want any specific things are there, I'll collect and I will mail you. Thank you very much.
Thank you, sir. Thank you very much for accepting our invitation in your busy schedule. We are very honored to have you, sir. I hope the participants felt very happy by attending today's session. And dear participant, you can ask any questions regarding today's topic.
Shyam sir. Hello.
Sir.
Vasantharaju sir.
Sir, Vasantharaju sir. Now our principal Manjappa sir is speaking. Vasantharaju sir.
Yeah.
Sir, our principal is talking to you, sir.
Hi, sir.
Dr. Manjappa?
Yes, sir. Manjappa speaking, name, sir. Yeah, Dr. Manjappa.
Okay, sir. Okay, sir.
Sir, nice meeting you. And thank you. And it is really need of the our topic. And actually, I'm happy to share that we also started working on some method development on using green solvents, as you discussing. And from management side and our institution stakeholder
this
side.
Is our pharmaceutical chemistry department.
Yes, sir. Yeah. So thank you so much, sir, for accepting our invitation. And my heartfelt gratitude for being with us today and sharing your valuable, time and your expertise in the field of
Thank you very much.
Green chemistry. And hope we will continue, collaborating on some topics in future. Sir, actually I have one query, doubt. Is there any research going on using green solvent for analytical purpose, like biosimilars or proteins, peptides, such kind of drugs? Hello,
yes,
sir.
Yeah.
Can you repeat, sir?
Yes.
Can we transform this, green chemistry, green analytical solvents for analysis of biological kind of drugs?
Yes, whether it's an analytical method or bioanalytical method doesn't matter. It is.
Can we shift to biological drugs, sir? Like nucleic acids,
so once
RNA,
you develop
mRNA, such kind of drugs can...
It is nothing but bioanalytical method is extraction from the drug from this one, just using the developed method you have to analyze. That's all. Yeah, maybe, but whatever the methods are available, only for the synthetic drugs are available. The same is in the biological analysis, you can do it, but biological samples, I didn't find much of the articles.
Yes. It seems unexplored areas, sir, actually. Most of the, biological...
Even whatever the derivation, you can combine the green chemistry with the green
analysis. Yes.
Something like if it is a... Yes, it is really, it is a, and I don't know, it is a future of the chemistry. Like you have a lot of innovations in the green chemistry. So here also you can go for, that means if it is a UV method is there, so your sensitivity is less in this one. So then same the green chemistry synthesis we can convert into a what is called the fluorescent compound. You can analyze by the green analytical chemistry. So that is that and all permutation combination you can do. Chemist, Department of Chemistry, you have the synthesis, analysis. So a lot of roles are there. You can say pharmaceutical chemistry is a mother
Yes,
of all
sir.
the pharmacy subjects.
Yes, sir. Yes, sir.
Is it not? Yes, sir. Vaibhavi,
Someone raised their hand. Vaibnavi Jambole. Please ask your question now.
Hello, sir. I want to ask here the good reference that you have referred for this procedure, because I have
Yes, sir.
referred
Yes, yes,
the detailed
yes.
data of that research paper of just me tell you the name, Francisco Pina Peria, that agri software data I have just reviewed, but still I don't understand how I can judge my method according to that principle. So I just want to ask you, so can you provide me any good reference paper? Because while listening to you, I get here some ideas, but still some confusion is regarding there in case of in situ measurement, or some problems are there still. So I just want to ask good reference.
Madam, I have some I told no for this to make this the presentation around 50 references I referred. So if you share your mail ID, I can share you the those references.
Okay, sir. Okay, sir.
So my mail ID
I will share.
share you. I think Dr. Shyam have my mail ID.
Yes, sir. I have. I will share to the participant.
Yes, please share to them.
I will share your mail ID to
Whatever
the
the
participant.
references
Now,
I have, I
participant
will
can
share
contact you, sir, through mail.
In that one, what is needed reference, you can
Yes, sir.
refer,
Yes.
madam.
Okay, sir. Okay, sir. Thank you, sir. Thank you so much. Nice session.
Thank you. Thank you. Thank you.
Dr. Vasantharaju, sir, it is a good and informative session. And today, sir covered regarding green chemistry, green analytical method introduction, objectives of green analytical chemistry, core green chemistry principles, several green chemistry, green solvents used in HPLC and other analytical methods, softwares and tools used to assess the greenness, and TOPSIS technique and its advantages. Thank you, sir. Thank you very much. And I have one, sir, here several faculty members are there in attendance
Thank you,
session,
Dr. Shah.
and scholars also there. Sir, can you, based on your experience, can you suggest the best operation, HPLC method and development? Can you give any tips to perform during the method development and validation for green chemistry? Sir, is it audible?
Green chemistry, again, there's a lot of these. Mainly, what is that ethanol and other pH. That is only greenness. No, your green chemistry means whatever the solvents which are eco-friendly solvents with that one and varying pH. With that only you have to try. Most of the methods
Yes,
uses ethanol.
So
and
during
varying pH. With that only you have to do several permutation, combination and all. So far, many green solids are not available. So here they have used ethanol and water, or ethanol and various pH combinations. With that only you have to go ahead. Yes. You are
Yes, sir.
the voice is not clear. It's breaking, stopping in between.
Yes, sir. Yes, sir. Maybe due to network issues, it is going like this. So, dear participant, you can ask any questions.
No, there is
Sir,
something
what
net problem
question?
is there. I think your voice in between is breaking, and I cannot.
Yes, sir. One more
Yes.
question, sir. During HPL
Manjunath was
simulation,
telling same I experienced.
yes, sir. During HPLC operation, and when so much acetonitrile solvent is wasting, na, sir, is there any solution to cut down this one?
Yes, yes. Cut down this one means one alternative is this greener method. Another alternative is that one reduce the runtime.
Yes, sir.
Reduce the runtime, and maybe industry methods are available to process the whatever it is they're coming out of this one. So usually, so what is that your institution level, what you will do will you will put the outlet to the sink, it will go to the sink. But that is
Sir, sir, is it audible, sir? Okay. Sir, is it audible now? Sir, unmute yourself.
yeah, it's audible now.
Yes, sir. Okay, thank you so much, sir, for accepting our invitation during your busy schedule. On behalf of our management, Vasantidevi Patil Institute of Pharmacy, Kodoli, and our beloved principal, sir, Dr. Manjap, sir, and participants, faculty members, research scholars, I would like to extend our sincere thanks for joining us and sharing a valuable session. And I felt the participants happy with the session. Thank you very much, sir.
Can you hear my voice?
Now it is okay, sir.
Yeah. Thank you very much. I'm thankful to Dr. Shyam, Dr. Manjappa for giving me an opportunity to speak on this one. So any participants, you just I'm ready to give all information if they contact me. Thank you very much. Have a nice
Thank
day.
you, sir. Thank you so much.
Can I leave? Can I leave now?
Yes, sir. Yes, sir. Please.
Yeah, thank you. Thank you. Thank you.
Thank you, dear participant. With this,
Thank you.
thank you. Thank you, sir. Thank you. So, dear participant, with this day 4, it is completed. Now, tomorrow we will meet in the same time. Thank you, one and all. Thank you all.